So I'd like to take the opportunity to tell you a little bit about LifeEdit today. We're a wholly owned subsidiary of a company called Elevate Bio based in Waltham, Massachusetts, who are very much an accelerator of the cell and gene therapy field. LifeEdit is a gene editing subsidiary, and we're based in RTP in North Carolina. So I want to take this opportunity to tell you a little bit about the gene editing platform that we are building down in North Carolina and how we're applying it. See here. So we've really built a very broad platform with a breadth of editing modalities, and we've done that because we want to be able to really think about the therapeutics we're going to address and make sure that we have the right editing tool available to us to be able to take on that indication. So we're focused on developing therapeutics, we're focused on building out our platform, continuing to stay at the cutting edge, and then we are actively trying to develop or establish partnerships and collaborations, and we've done a number of those in the last 12 months. So it's been exciting times. So as I said, we've built a broad platform, so this isn't just a single editing modality, and it's all been built on the back of our nuclease platform, and I'll talk a little bit over the next couple of slides of where we've identified those nucleases. But they are the foundation. We've been able to show that we can do knockout, insertion, and then I think, interestingly, we spent time pretty early in the company timeline building a proprietary deaminase collection, and so we also have both C and A base editing available to us, and we're starting to build an RT editing platform behind that. So where did our nucleases come from, and what makes them interesting or different or whatever, but differentiating? So we really have identified a large number of distinct nucleases with a very broad PAM diversity. And so the work was originally done out of a collection we have exclusive access to. It was a microbe collection that had been fully sequenced. We did the kind of standard bioinformatics identification and realized that there were hundreds, thousands of these systems within the collection. We focused down on the type 2s and the type 5s and started to do the wet work to understand if these were going to be active in mammalian cell systems and beyond, and they are. So at this point, we've got this large collection of RGNs, as we call them, and they've got, we can pick and choose which ones we want to use based on the PAM that we think is most appropriate. So this is where we started in identifying the nucleases. We've actually subsequently moved on, and we've started to think about it from a kind of broader perspective and doing a little more work in the metagenome and starting to expand our database. And so at this point, we've got access to close to 800,000 CRISPR systems. They cover all the Cas subtypes, and there's billions of proteins out there that we're continuing to mine and continuing to characterize. But we're not here just to collect nucleases. We really want to identify those that are going to be interesting to move forward and to think about in a therapeutic context. So we have 4 lead nucleases that we've identified over the last couple of years. What you'll notice is that they have orthogonal PAMs, so that's one of the reasons we've kind of focused our deeper work on these 4. They're called LEGs for life edit genes, and the other thing that's noticeable about them is that they're smaller than SpyCas9. This was that was one of our early filters. We wanted smaller nucleases. We wanted to be able to package in AAV. And so we went after those specifically. We can add in the guides. We can add in the other regulatory elements we need. And we've shown that we can package them in AAV. And we have also shown that some of our base editors are packageable in AAV as well. These are all proprietary, they have low homology to established systems, and we have a robust IP strategy protecting this and the rest of our platform. So we have this collection, we've built the nucleases, we've built the base editors, and we wanted to start thinking about how we were going to use them. So we, like most gene editing companies, think about these from an ex vivo perspective. We're enabling cell therapies as well as applying them as in vivo gene editing therapeutics. I'm going to focus the rest of the talk really on the left-hand side of this slide and show some of the data that we've been generating in this space. So, these nucleases can function in knockout as you would expect. So as you look at the left-hand side of the slide, you can see editing. This is just straight-up knockout with leg14 nuclease. You can see mRNA and RMP delivery and straight knockup and high efficiencies. When we're initially thinking about how we're going to apply a gene editing system, we scan the guides, we scan across looking at the edit we want to make, and we can show that we can get highly efficient guides that not only show the editing we want, all of our editing is NGS, but we can then show the functional knockout. We've done a lot of this work in T cells, that was on purpose, but we've also Elevate Bio has a regenerative medicine team, and we've been fortunate that they've been able to leverage our gene editing systems and generate a significant amount of data in iPSCs. And again, they edit robustly, and those edited iPSCs can be taken on. We're able to do HDR, and this is very efficient. So this is work that we did, again, in conjunction with our colleagues at Elevate Bio, where we're doing insertion of a transgenic TCR into the track locus. It's just over 4 kilobytes, KB, and we get robust knock-in. And you can see this in the two graphs on the right-hand side, greater than 80%, and we've done this over and over again. So we're pretty happy with our knock-in capabilities. And then once we had been kind of getting the knock-out and the knock-in efficiencies up, we started to think about multiplexed editing. And so this is work that we were doing with the nucleases. And as you can see, we can get pretty efficient knockout of both TRAC and B2M. But as we were doing this work, we started to recognize, as everybody in the field is recognizing, that doing this with nucleases isn't necessarily the best approach. And so we wanted to think about how we could multiplex our base editing. So as I mentioned at the beginning, we have a proprietary base editing platform, both A base editors and C base editors. We've been using our C base editors predominantly in the cell therapy space, where we're doing multiplexed knockouts, and then focusing our A base editors towards the in vivo therapeutics. And so the way we go about it is we start with our C-base editors, make sure that we're getting the initial, identifying highly efficient guides, then going ahead and multiplexing those and optimizing once you're doing it all together, and then finally doing the functional testing to ensure that what you think you're doing, you're actually, you're getting the readout you want. And so this is an example of a 3-plex editing that we did with our C-base editors. These genomic loci are loci that were of interest to our cell therapy team, and so that's why they're masked at this point. But you can see that we get good editing in the single plex, greater than 80%, and we retain in the majority of the edits the similar levels of editing when we multiplex. Most importantly, you then see the protein knockdown. And we've gone on and shown this not just with this 3-plex, but with a number of other multiplex systems. We've also done it with 5 genes. Again, this was for a project that we were doing internally, and again, we show that you can get similar results. And so we think that this is quite robust. It takes some work to get the right guides in the first place, but once there, we're able to move it forward pretty efficiently. And then finally, we wanted to push the limits of what we were doing and really leverage the fact that we have this unique PAM diversity. And so what we've been able to do more recently is show that we can do a simultaneous knockout and knock-in. We did this with an exemplar, so with the CD19 CAR expression and B2M knockout. And so what we're doing here is we're using the our C-base editor to do the knockout of the B2M, and then we're using Leg95, which has an orthogonal PAM, to do the track knockout and insertion of the HDR cassette. And so what you can see is we get very high levels of knock-in and knockout simultaneously. And what we're also trying to do, or we're actually doing, is we're doing this in conjunction with the manufacturing team at Elevate Bio, because what we want to be able to do is build a system that is kind of a robust platform that you can integrate into your cell therapy, and that you're doing this gene editing at the same time as manufacturing your cell therapy, so that the timelines match up, and so that you're really exerting as little pressure on these cells as you need to do. We know that if you do this stepwise, the T cells, they don't like him as much. So what we've tried to do is build a platform where you can pick and choose what you want, you can pick and choose the tools you want to use for the edits you're trying to go after. All these gene editing tools have pros and cons, and it's about choosing the one that is going to be most efficient for what you're going to do. It would be hard to have a conversation without mentioning the off-target analysis. And so again, this is quite specific to the program that you're running, but we've tried to set up a pipeline that starts from the very beginning, so that when we're first doing the guide streams, we're looking bioinformatically to understand what the off-target profile is likely to be, eliminating those that we don't think are going to be suitable, and then continue to build in the complexity of the off-target pipeline as an asset moves forward. We've done that with a multiplex edited T cell where you've got 2 knock-ins, and we were able to identify guides that were going to be both efficient and have the off-target profile that our partner was looking for. So I'll finish just by mentioning we do have an in vivo gene editing therapeutics approaches internally. We have an internal pipeline. We're focused on diseases mainly of the CNS, the eye, and the liver. We have announced one of those publicly, which is Huntington's disease, and we're starting to bring in delivery there with our AAV and an LNP platform that we have in-house. And that, I believe, is my final slide. So open to taking any questions. Or not? Yes.
Was this done by transfection? Excuse me. How was this done?
Yes. That is, the C-base editors are delivered as mRNA.
Delivered as mRNA, and
Yeah.
this is in what cell type?
That was all in T cells.
This was in T cells?
Yes.
Okay,
Yes,
all right,
yes.
great, thank you.
Yeah.
C-based editors, what's the window of editing? As you mentioned, it's limited often. If you have Cs that are adjacent,
Yes.
it'll deaminate those as well.
Yes, so yes, we obviously, yes, you're absolutely right. You've got this, there's a lot of taking a step back, yes, you're absolutely right. You've got to get your context specificity pretty tight. You've got to be focused on the edit you want to make. You can get bystander edits, but we really spend time designing, making sure we're using a nuclease with a PAM that gets us where we want to be, but doesn't affect the other Cs that are there. It's the same with the A-base editors, and then using the guide to kind of modulate where we're going. So yeah, there's a lot of screening involved, a lot of optimization. We have a whole team that's also engineering the nucleases and the deaminases to try to reduce bystander edits as much as possible.
Is it a simpler window to David Luke's base editor? Like
Yes.
6 base?
Yes, yeah, absolutely, absolutely. Yes, all of this work is clearly on the foundation of the work that's been done by others. Excellent. Well, thank you.